Good afternoon, everybody. It's nice to be here to speak with you. I hope I can give you some new ideas about a product which is not really known in nutrition. These are so-called nucleotides. Now, when we look on a When we look on a proper nutrition should really go to give an optimal performance. It should be an optimal body composition. You should have less fatigue and a higher level of energy. The recovery should be better, and mostly you should have a better immunity. This means less inflammation, less soreness, and the risk of, let's say, injuries should be lower, or a better immunity, and also the ability to perform sport should be improved. We base everything when we speak about proper nutrition on proteins, fat, carbohydrates, minerals, vitamins, but we forgot something. Because why we are not reaching these goals? Because this is what you want. You want to have all this, and you don't get it all the time. And we are speaking about nucleotides as a forgotten nutrient. Because all the recommendations, as I mentioned before, are based on proteins and so on. But nobody really looks on the recommendation on nucleotides. But nucleotides are really the basis of all this. And when you look with afterwards how much nucleotides you get in food, you will be surprised. Now, what should we expect from nucleotides? Also, the nucleotides are the basic of all cellular development, and they are really important for the reactivity of the immune system. And the reactivity of the immune system is of most, even for sport people, is of most evidence, because you have to see, for example, a cell multiplication in an immune system takes between 8 and 12 hours. One cell doubling. A bacteria doubles in 20 minutes. So when you have, let's say, one bacteria in the body, after 20 minutes you have 2, then 4, after an hour you have 8, 2 hours you have 64, after 8 hours you have 64 million, before one immune cell can divide. This means the reactivity of the immune system is very much depending that they get everything ready. And what is the problem is that the nucleotides are found in really low levels and low bioavailability in our daily food, like vegetable, fruits, meat, and so on. Highest concentration, I will come back later on this, is on what we call offal. Also intestine, liver, lung, everything we throw away. We think it is no value. No value, but this is from nutritional part is the most important thing. When you look a lion or whatever you see, when he kill an animal, what is he eating first? He opens here. And animals are clever. They know what they need. And we will see afterwards a little bit what this means in this direction. And nucleotides need not only be present in a certain level, but also in the right form and in the right proportion. So we call the nucleotides the best kept secret in nutrition. And it is quite interesting when you look on how the effect of nucleotides was discovered. It was discovered because when mothers started no more to breastfeed the children, but take these milk formulas, let's say from Abbott or whatever. Children had a bad immunity, a lot of immature lymphocytes, they have a bad digestion, and then they have a bad brain development. And what when then looked what is the difference between these milk formulas and mother milk, it's mostly the nucleotides. Nucleotides were extremely high, whereas in mother milk, whereas in cow milk it's extremely low. So this is the reason why we call it really the best kept secret in the world. Now, what are nucleotides doing? For what are they here? Also, because when we look what our body is doing, we have a constant flow of cell. We have a formation and a degradation of cells every day, millions and millions and millions. And the nucleotides are both the building blocks of DNA and RNA. And here, I will come later, RNA protein synthesis. And here, then you see that the nucleotides are not only in the cell multiplication or protein synthesis, but in a number of other metabolic processes. By the way, if you have any questions or if you have any remarks, please interrupt. Feel free. Now, I just go quick into more biochemistry part, not too much. But you see that this is a DNA. It's a double helix, which is made up from nucleotides. The nucleotides is guanosine, cytosine, adenosine, and thymidine. So they build, I show you later, they build this long chain. And from this, you need this. These are always, let's say, you have here the guanosine, cytosine, here this is the thymidine, and so on. So it is a long chain. And the same is true here for the RNA. But in RNA, first of all, the sugar is different. But here, instead to have thymidine, you have uracil. So you need, in fact, for a proper nutrition, not 4, but 5 nucleotides. Now, when we go a little bit again in basics, all cell in the whole realm of creation, cells are always the same. They have a cell wall, they have a cell nucleus, and in the nucleus you have always the DNA or RNA, and they can produce the nucleotides for the potentially themselves. Potentially, I say. And then the nucleotides are part of the genetic information, but every cell, also the DNA of every cell, consists of 8 billion nucleotides. 3 billion, sorry. 3 billion nucleotides. I'm exaggerating. Start to exaggerate. Now, to have the production of the DNA, the new cell, it's always start with the doubling of the DNA. This means the doubling of the DNA needs these 3 billion nucleotides. Otherwise, it doesn't work. But in this process, you also need the protein. And the protein, for the protein synthesis, and I come also later on this, you need 5 different molecules of nucleotides. This is messenger RNA, tRNA, you need the DNA, messenger RNA, tRNA, ribosomes, and energy. And the energy is transported only in form of nucleotides. Then you have additional effects, as I said before. We have this energy. It's as a second messenger. This is cofactors in enzymes. We have in the body about 50 to 100 enzymes, which has a cofactor made on nucleotides. And they are really important for fat, carbohydrate, and protein synthesis and degradation. So again, nucleotides. And at the end, as I said before, it's the effect on energy in the body. And just quickly, what is a nucleotide? The nucleotide is made on a base, and the base can be either a purine. I go a little bit. Can you see it over there? A purine or a pyrimidine. We have 2 purines. This is adenosine and guanosine. And we have three pyrimidines, cytosine, uracil, and thymidine. And you see, here we have the nucleobase, and when it is connected with the sugar, and this sugar can be either ribose or desoxyribose, you have a so-called nucleoside. I say it purposely because I come later. And you see, this is only base and sugar. This is a neutral molecule. It's very important later. For the nucleotides, monophosphate has a phosphoric acid attached. And then for the ATP or UTP, you have two other phosphoric acid. And here you have the energy in the binding. So the binding is in this form, and so in nucleotide 3-phosphate. Now, the whole connection in the body, also in the DNA, is always you have the sugar part. Just a moment. When you look here, this sugar part is connected over this with the bridge with the phosphor part, then sugar and phosphor and sugar, and always the side chain is the single nucleobase, either guanosine or and so on. And this, so here it's again just an overview of the nucleotides, and this forms then the long chain of DNA or RNA. So the nucleotides are really so the basis for all metabolic processes in the body. And we have seen we have energetic, metabolic, catalytic, and so on function in the body. And we have a constant need for nucleotides over whole life. Because you see here, for example, when we have a cell multiplication, the cell multiplication starts always by dewinding of the double helix, and along each of the strand, another strand is built. Also, for each nuclear cell, we need 3 billion nucleotides only for the DNA doubling. You see here, you have, and you cannot replace one nucleotide by another. When you replace a nucleotide by another nucleotide, you have a wrong information in the body. This can either lead to the death of the cell, to degeneration, or even to cancer. Also when wrong nucleotides are in the wrong place, and there are studies showing that a lack of nucleotides can lead to cancer. Also it's not something with, that nucleotides promote cancer, it's inverse. The lack of nucleotides can. And you see this whole process here, this whole process takes about between 8 and 12 hours. When we have here the double set of DNA, then it starts to divide this on two ends of the cell, and you see they are pushed outside, and then the cell starts to divide in the middle, and this whole phase takes only 20 minutes. So the longest part in the whole cell multiplication is really the building up of the nucleotides. And this is where we can do something. Now, because here I would like to show you how you can get nucleotides. And so I have said first, nucleotides can be made on de novo synthesis. But for example, the purines, you need 14 biochemical steps, and you need about 1 billion of purines. You see, when you have 14 biochemical steps times, you can make your own calculation how much time it needs. Pyrimidines need 10, so this is really a time and energy consuming process. The next process is here, so-called salvage pathway. In the salvage pathway, this is like a recycling of the nucleotides from damaged cell or dying cells. But this is only about 60 to 70% can be really recycled. The rest has to be, and here only you have what only when you have dying cells, then it can be recycled. This means, for example, in a growing age from children, they have nearly no dying cells because they are still growing. This means for them the salvage pathway is relatively low. And then you have the third one, which is the diet, and this goes into the nucleoside, and then you have a pool here which can be distributed in the body. So this is the way. So we have three, and this shows you also the importance of the nucleotides, that they have no other system have three mechanisms to get the for example amino acids you have just in food. Now here you have the own synthesis, you have the recovery from dying cells.
How much liver do you need to eat to get a correct dose of nucleotides?
Can I show you later on? Because it is I come here then, because this is I said before. So this salvage pathway is only 70 to 80% effective. Then the own synthesis is, as I mentioned before, it is really time and energy consuming. And now something very important, extremely important, is that the cell of the gastrointestinal tract, they can produce nucleotides, but they can never cover their own needs. And they can get the other nucleotides only through food. The cells, all the cells coming out of the bone marrow, white and red blood cells, so the defending cells and the red blood cells, they are not able to produce nucleotides. They need it from outside, and the only organ in the body to produce more nucleotides for their own need is the liver. But in many cases, the liver, you have, let's say you have a disease, then the liver function is always going down. So I mean, in a time where you have the highest need, the production is the lowest. Also, sometimes when you have, let's say, high sport, a really intensive sport, liver function is going down. Then you have less energy, less nucleotides available, and therefore the performance is going down. And then blood cells, as I mentioned before, and here I did not mention bifidobacteria. For some bacteria cannot produce nucleotides. And then here it is absorbed from the food, and now here the content has a lot of variation. Food has a huge variation of nucleotides, and also the bioavailability is extremely low. It's only about, from food, you can only about take between 5 and 15%. The rest is not used in the body. So you see, external supply can save energy in raw material, support active processes, and speed these processes, also the recovery of the body. Now I would like come to your questions. I would like to show we have made about, all in all, maybe 1,000 different analyses of food and looked at different food ingredients of food. And you see that we have in vegetables and fruits, the number of nucleotides is extremely low. And when you look here, you have, for example, broccoli, which is pretty good, the total nucleotides. Also cauliflower is pretty good. Now something which is here a problem, that we have only this much of purines, but we have so much of purines. This means the usage for really cell multiplication is only down here. Because you need all the different nucleotides in the same level. For this. That's good. But you see, you have, and you see every time that the pyrimidines is low and the purine is high. Now you see here there are some. Here is, for example, spinach is not so bad, But are you go on? But when you look now, let's say meat, and when we look here, you see that the scale is the same. You find here high proportion of nucleotides. What is this? It's all intestine. This is the small intestine middle part, small intestine lower part. Here is, for example, kidney, lung. This is the large intestine, tongue, brain, heart. And you see in heart, it's quite interesting, you have nearly no pyrimidines. This is also a reason, because the heart is a pump. And you need there a lot of ATP, GTP. This means all the purines are high there. Everywhere where you have high energy needed, you have high purines. Everywhere where you have, let's say, more or less a cell multiplication, I come later on, you will see that you have a more balanced ingredients. This is same with pork, and you see the same picture, absolutely the same picture as before with beef. Now here I have just different food, and you see, for example, mushroom, okay, it's not bad. Also watercress is not bad. Marmite, but you see with Marmite you cannot balance the feed. You can have nucleotides, but you have the same as in the other food. So what we want to do is to inverse, to put the pyrimidines up and the purines a little bit down. This is what we wanted to do. And it's quite interesting when you look, for example, white fish or mackerel and tuna, it's extremely low. And steak are down here. It's extremely low. And here is blue cheese and so on. And so, except of what we throw away, nothing really contains a lot of nucleotides. It's amazing what we are doing in our nutrition. And here, just again to show you a little bit organ meat, because this is liver and so on, and vegetable. You see, really the high number of nucleotides you get out of, let's say, animal products. Now, I was telling you that we need for every cell 3 billion nucleotides. In a day, 2 billion blood cells are replaced. 2 billion blood cells. Oh, by the way, I forgot your question.
The question was, how much liver will one need to eat in a day?
So you need because we have to look a little bit. We have to look a little bit into this one here, in the pyrimidines. You need, I would say, more than a kilo.
How can we have that requirement? It's not, we couldn't satisfy that naturally. So
this is exactly what is the problem, because when you have, let's say, a normal life, easy life, they're all in the old age, they were not hunting every day. They were not hunting the elephants and so on. They were hunting the easy animals, and they went hunting only when they needed for feed. So they had an easy life, and for them that was okay. We are now on in the same pace. We are stressing the whole day. In the beginning, morning, when you get up, oh, what I have to do this. We're already there, 200% of any of our performance. And what we did in the meantime, we improved all our food with protein and so on, what I said before, but nobody thought of nucleotides. So you can still perform, but you don't perform as you always get sick, you always get tired, you can do it, but you cannot do it under optimal pace. That is what I wanted to say. Oh yeah. So you see, only for the blood cells replacement, you need 6 billion billions. How you call this? Huh?
A lot.
A lot. Yeah. And then normally, compared, this is only for cell multiplication, not for protein synthesis. The amount of RNA in a cell is 1,000 times bigger than the DNA in a cell. Because these items are constantly new made. And then you need this information in the body. You need the enzymes, which are made of, and you need the energy. You see that the need of nucleotides is extremely high, and we don't think on it. And now I just give you a little bit an overview of the lifespan of the single cell. The only remaining, the only part of our body who is lifelong is our brain. All the rest is replaced. And so bone cells are quite long. It's about 20 to 30 years. This means if somebody is 50, you have the whole body is, 30, the whole body is new, except of the brain. The muscle cell still about 10 years, fat cells only 8 years. Here, the lymphocytes, it depends on which one, again, can stay over a year, but the liver is only about a year. In a year time, you have more or less the whole liver remade. Then red blood cell is 120 days, skin cells only 30 days, column 4 days, intestine 2 days, stomach 2 days, and the blood neutrophils only 2 days, 1 or 2 days. So, in fact, we can say every 5 years, we are new. We are a new person. It's amazing, eh? Here. Also here is just a summary, because I find this so amazing for me. Now, what we did, we made a supplement, and this supplement, we tried to put this down, this extremely low, because these of what we have seen is already in food. But we put this here, this tree, we would create it quite high. And in this way we cannot 100%, we try to balance the whole, let's say, imbalance of the feed, of the diet which we have. So this is, and we put it in a way that it is really easily accepted in the gut. Also because we purified these nucleotides, and when you have purified nucleotides, the uptake can be between 80 and 95%. Also nearly everything goes into the body. So we can, even with relatively low amount of nucleotides per day, we can balance what is missing in the food. Now, I said before that cells are able to produce nucleotides. We have exceptions. This is everything what comes out of the bone marrow. Lymphocyte, macrophages, red blood cells not able to produce nucleotides. The intestinal cells, they can produce nucleotides, but the needs, and you have seen before, it takes only for about 5 days for the cell to be replaced. Their need is so high that their own production can never cover these needs. So they need it in the feed. Then here, some cells of the intestinal flora, like bifidobacteria, cannot produce nucleotides. So when we feed, we did mostly animal trials. When we feed these animals, we found always higher bifidobacteria and a group of bacteria which is immune stimulating. That we have also seen. And then some of the brain cells, because in the brain, it's not only the cell which is important, also the connections between the cells also come. And this is, again, depending on the protein synthesis. And now here, I mentioned before that these bone marrow cells are not able to promote, also really to build the nucleotides, so they are fully depending on outside. And therefore, we first of all, because otherwise the liver has to produce it, and then we save energy and also the precursors of the nucleotides. And we have seen that when we give nucleotides, we have really an improved immunity. And now the immune system and performance are connected very much. The immune system is under a lot of pressure. You have, let's say, for example, most of these are negative effects. You have mycotoxins. You know mycotoxins? They are toxins out of food, which, for example, aflatoxin is one of these mycotoxins. You have the climate. Depends when you have, let's say, like yesterday, this cold weather, I feel half sick. You have bacteria. You have stress. You have pollution. You have viruses. The only way where you can positively influence the immune system is by nutrition and clear your mood, so your brain. But here you have an effect on a positive effect on the immune system. And when you look what happened on the performance, when you have, let's say, a stress factor to a damage on the immune system, this means this leads to an immune suppression, and immune suppression always leads to a poorer performance. This means you have to care about the immune system to have an optimal performance. I have to look a little bit. And it is extremely important that we have a balanced diet to overcome all these problems, because, as I said before, the bacteria, they can produce the nucleotides themselves, and they are very quickly developing. And when we are not keeping pace with what happens in this bacterial growth, then we are lost. And we have done a trial with animals. We have stimulated these animals with so-called lipopolysaccharide. This is a part of the bacterial cell wall. And this is really a strong immune stimulator. And we looked how much it takes to double the IgG level in the body. In the control group, we needed 8 hours to double the IgG level. In the group where we have really high amount of nucleotides, we have in 2 hours more than after 8 hours in the control group. And I made before this calculation that after 2 hours you have 64 bacteria. After 8 hours you have 64 million. So you see the immune system is much easier can much easier overcome a problem with 64 bacteria than 64 million. And this is what we want to do with this. And what I want to say here is that the efficiency of the immunity really depends on your nutrition. Yes.
Have you been able to do? Have you been able to test it on people?
Yeah, I have here just this was a trial. This was a trial which we did in Edinburgh, and we looked here people coming with a cold. Also they were students. They came with a cold into the institute, and then they had to fill in a questionnaire, and they measured also, for example, some of the symptoms. Also they also measured, for example, IgA in the mouse. But here, for example, these sufferers, and you see in the placebo, when they came in, they were already, they went even up here. They went here up, and only after about 7 to 10 days, they were overcoming the problem. When they came in and immediately got the nucleotides, they were after 5 days it was gone.
Was it a supplement?
That was a supplement, yeah.
And that was created in a lab?
No, that was the product which I mentioned here before. this year, where we made this special composition that we have, let's say, lower purines and higher pyrimidines, because this is the composition of the nucleotides in a supplement is extremely important, because you see before from the nutrition that you have always purines high, pyrimidines down. We did it inverse. We put pyrimidines high and purines low, and in this way we try to balance a little bit the effect. Sorry about that. And here, for example, there was different study which we made also. For example, here, these were two studies we made with Professor McNaughton. This was moderate endurance exercise and a high endurance exercise. And we found here that the IgA in the mouse was 10% higher than in the control group, and here 20% higher in the control group. This means because when you have a high exercise, the immunity in the mouse, when you really have a high exercise, goes 90% down. So when you can avoid this lowering, because you can only get the infection either to the mouse, to the or to the skin or to the lung. And this means otherwise you are protected. So normally when you have an intact skin, you can go in a mud, nothing happens. A little cut, you will have an infection. This means you have different immune system there, including IgA on the surface, which neutralizes bacteria and viruses. But you see here, this is the effect on antibodies in humans. And then we have also looked. Here is just again this that we have here another trial which was 25% higher with nucleotides, and here is the other effect on, because this is mostly on immune cells. Here is the effect on red blood cells, and this was a trial which we did in horses. And horse trials, also animal trials, are for me always more precise than human trials. Because in animals you can keep them extremely identical, you can treat them identical, you can feed them identical, you have the same genetic, you can look that they have the same performance. And here in these trials we measured, in horses, we measured the red blood cells. And you see in the untreated, we have nearly no, after 6 months, still, 4 months, still a little bit the same. And here we have a significant increase of red blood cells. And also the hemoglobin is going drastically up. And this we measured here in another trial. And just here, this trial was so important because we had here these animals were absolutely trained and kept the same. And we have 5 different periods. And always at the end we have a standardized test. First was acclimatization, then we have light training, 3 weeks, we have aerobic training, 3 weeks, anaerobic training and detraining. And what happened here, we looked on the oxygen consumption. You see, we have put always the first day, also the number of oxygen uptake as 100%, and you see we have in both in the light training, we have in the same level, but afterwards the oxygen consumption in the control animal went down, and only here it went up. Whereas with the nucleotides it went up drastically. We have more oxygen uptake, even in different speed, and even when we calculate this CO2 expiration, it was much higher than in the control group. This means these animals had a better were better, how you say, supplied with oxygen. And when you have more oxygen, the degradation of lactose is different. And you see, this is just again this trial. This was the heart infarct frequency was lower, but you see the lactate, instead to go up or stay on the high level, it even went down. And this again, acclimatization, light training, aerobic training, anaerobic training, and detraining. You see, and this is exactly what people want, that you stay longer in an aerobic situation. Your body stays longer in an aerobic situation. You have less damage. And this is quite interesting also, cortisol. Cortisol is like a stop signal for the immune system. When you have an increased number of cortisol, then the immunity can drop drastically. And you see here in the control group, we are more or less on 100%, but here we go about 15% lower, even under high training. Normally you see the inverse effect. This is after work, the same. And then this is an important parameter because GOT measured damage in the cell and in the liver. When you have this enzyme, you find normally within the cell, and only when you have cell damage, you find it in the blood. And you see in the control group, it goes up about 300 times. Whereas here, we go up about 60%. So we have much less of muscle and liver damage with the product. Now, in the human field, again, we look here in different trials. This is an ongoing trial in Liverpool, and this was the trial which we did with McNaughton. We saw here the impact on recovery. This is blood lactate again. We found here in the placebo, in the trial, 23% lower than in the placebo, and here 25% lower than in the placebo. And both were high intensity activity trials. This one here, the one was this repeated sprint accommodates. You see, when you look here, we were at this level, and afterwards down. Whereas when you look in the placebo, it went up, and here went down. And this is exactly what you want, because lactate is also connected with cramp. Then here, the immune suppression again with cortisol. And again, this trial here, we have 10% less cortisol build up. And here we have, in this trial, we have seen, this is just new, this is coming out now in 2016. We have 25% less build-up of cortisol. Here, this is the effect again, a little bit better shown. Now, here it's something quite interesting. I normally have a line in here. I can put it like that. You see, this trial, which is a new one, we measured this is again cortisol. We measured before the exercise, immediately after exercise, then 15, 30, 60, and so on, a minute later. And when you look here, and the same people were once placebo, and once were the trial group. And you see in the placebo group, you have an increase, a significant increase in the first hour, whereas when you look at the same level, at the same time, over in the group with the nucleotides, you see absolutely no increase, even a decrease here. So you see, this is quite an important. I think I took it here. This is very important so that because cortisol on the long run will really have will have a bad effect. This is very important mostly for people who are constantly training. When you do it once, cortisol is not damage. It's even good for the short term. but constantly, all the time, you will have a huge, huge, huge damage in the body. For
Why
the
does it drop away now in 2 hours?
oh, that is, we don't know yet what is exactly why, because it dropped really down very drastically.
Yeah.
Very drastically. I don't know yet why it comes, because this was the first time, because before we measured always one time after the trial. Here we would like to know what is the, let's say, the time curve of and this is something which we have not seen up till now. We don't know. Now, something very important also is the gut for people. And we have done trials in animals also, this because we have to make section of the gut. And we looked on the villi, and when you look here in this picture, the villi were 25% higher. Then we have clearly a bigger surface, but very important, when I don't know if you can see it really from behind. You have here an extremely good, around the blue, we have a very good so-called mucosa layer. A mucosa layer is full of antibody, catching all the bacteria in the gut and neutralizing them. Whereas when you look here, the mucosa layer is more or less gone. This means here you have a much better protection. Bacteria, and I told you before, when you go in a mud with your hand and you have an intact skin, nothing happens. As soon as you have a small cut, you have an infection. This means here, when you have, let's say, little cuts in the gut, you get chronic infections. And that is a huge problem. And we have seen here an extremely good effect. And different, so this was only one animal species. We have done in all kind of different animals. And we have always seen absolutely the same effects. And in this trial, this was a trial now where we looked in irritable bowel syndrome here in London. We had here 37 people, and we looked, they have to rate it all the time, every day, their symptom between 1 and 7, a score from 1 to 7. And we looked for abdominal pain, diarrhea, urgency, incomplete evacuation, flatulence, and so on. And what we have seen here, and this is quite interesting, we have a significant improvement of abdominal pain, we have less diarrhea, less urgency, and significant less abdominal pain. These three were also improved, but not significant. And most probably, when we would have taken longer over the time, because you see here the baseline before was more or less straight on. The placebo was more or less straight on a straight line, whereas with the nucleotides over the time, the whole symptom improved. This means the longer we could use, the better it would be the healing process. And this was a little bit the remark of the researcher. Also, we have this improved, and most probably because of the small number of participants, and when we would have a longer, let's say when we have more people and a longer supply, we will have most probably all the different parameters improved. So we have here really an implication over time. And it is also quite interesting. In most of the trials we have seen that the amount of nucleotides is important. When you would like to have a very short and quick reaction, you need absolutely high amount of nucleotides. Then you get really a quick response. When you, for example, you can distribute it over a long time, you can lower the intake. But the very quick response, you need high amount of nucleotides. Here I mentioned already that we have this effect on bifidobacteria. Now we have other roles of nucleotides I mentioned before. Here energy transport is everything is on nucleotides. this is, most people do not know that when you have a hormone release, the hormone comes on the cell wall on the wall of a cell. But internally, you have a whole process, and then this message goes over, and you have a building up of so-called cyclic AMP or GMP. And this really has afterwards an effect on the DNA production, also protein synthesis. So nucleotides are what we call second messenger for hormones. Then very important here, this is the wound healing, because wounds is always a big issue. We have the first effect in the wound healing is the macrophage. Macrophage is going to clean the wound because you have a lot of cell debris, and when the wound is clean, the macrophage release some hormones or some signal molecules to activate the surrounding cells to multiply. So here the number of macrophages, and I told you before, macrophages are depending on outside delivery of nucleotides. And here it's something very important, this DNA, because the protection of the DNA is most important. And we have done here a trial where we have here the energy came mostly of starch, whereas in this group we have replaced starch by linseed oil. And linseed oil is quite high in PUFA. And PUFA can release an oxygen burst, and they damage the DNA. And here in this group we have done the same, but we have added nucleotides. And what we measured, we took the DNA out of the lymphocytes and put it on an electrophoresis. And the DNA is about this long. When you put this on an electrophoresis, it will not move, and the whole stain will stay here. And only when you have, let's say, smaller parts, when the nucleotides are cut, so the DNA is cut in smaller parts, you find here a tail. And what we have seen here is in the beginning, in all three groups, we have about 96% intact DNA. After 21 days, we found here 96 still, but here 85. This means there are 11% of the more damage than in this control group. When we put in the same diet nucleotides, we are 95%. This means even damage in the DNA can be repaired, and this means that you have less cell who has to be replaced. And here you see the picture. You see this is the control. This is the oil, and you see here this tail. Therefore, this trial is called comet test. It looks like a comet. And here oil plus nucleotides. And now the protein synthesis, again, very important. We have, I told you, 5 different nucleotide items needed for protein synthesis. You have the DNA, which contains the information. This information is loaded down on a new made messenger RNA. And for one protein synthesis, because you have not only one, you need repeatedly making up of this messenger RNA. They go on an organelle called ribosome. Ribosome is 60% nucleotides and 40% of proteins. And here they line up, and then the so-called tRNA, again nucleotides, bring the single amino acid to the place where they are connected, and the connection needs energy. 5 nucleotide items for one protein synthesis. And people do not really realize how much nucleotides are really important for the protein synthesis.
How do they react with heat? Like example, cooking. Do they get destroyed due to heat or?
Excuse me?
Like you said, they're quite rich in certain foods.
Mm-hmm.
Like you mentioned, a bit awful. But how do they react to heat? So when you cook them?
Also, we have done quite a lot of trials in animal feeding, and animal feed are mostly extruded at 160 degrees. And we have put it there, we have no losses.
No losses?
No losses. We have put it in an autoclave because we want to put nucleotides together with the vaccine. 121 degrees for 20 minutes, no losses. So they are extremely stable.
Yeah.
And even when you look let's say when you some of the old animals from the ice area, you've still found nucleotides in their bones, for example. Now here is a here is a trial with which we look what is then afterwards on the performance, and this is an ongoing trial. This one here was a repeated sprint exercise where I had 6 times 35 meter in maximum speed. Then they have to go back in 15 seconds, and then the maximum speed. The average speed here in the placebo was 7.06 seconds. With the nucleotide, it was 6.6.
Same athlete.
Same athlete. And in this part here, I showed it here again. This was again this exercise, this very heavy exercise, and you see the peak force in the control group. This is afterwards, immediately afterwards, 24, 48, and 72 hours. Yeah, you see in the control group they recovered after 72 hours. In the group with the nucleotides, you have no losses. So for me, nucleotides are really something for athletes, which is, for me, just a must. If you really want to perform an optimal, and we have people in Switzerland, for example, we support how you call it shot putter. Yeah, he was really sick, and we gave him the product, and he said the force he has was higher than the actual world champion. So he just has to work on his technique, yes. But the force, he has more than the actual. So, as a summary, we say nucleotides are essential, mostly on a certain disease period, when you have limited intake, like say when you are vegetarian or even vegan. Normally they have problems to get nucleotides. When you have heavy exercise, and period of elevated stress. And this is not only related to sport, it's even mental stress. You will have the same effect. So, when we give the purified nucleotides, we can reduce the efforts to produce in the body. They still produce. We are not cutting down their own production. We give just what they need more. And we accelerate most of the activated process, and we save a lot of energy for the body.
Can your liver produce nucleotides from fatty acids?
No, nucleotides are...
Convert.
No, that is not possible, because they are totally different molecules, totally different molecules. So, yeah, okay. Is there any questions?
Where do you source your nucleotides from?
You can source nucleotides from every cell. But you need to really to make good source of nucleotides. We use yeast, but the special grown yeast, because
you're not using loads of cow intestines or
no.
That's what I was concerned.
No, because I know from a company where they produce nucleotides from intestine, that is always a little bit a problem, because you have to be extremely careful with contaminations.
Yes.
And this is the reason why we purify the nucleotides. So the whole process is that we extract first RNA and DNA, and we separate, let's say, the cell wall and the rest. And then you have to hydrolyze the nucleotides. And then you have to put it on an ion exchanger. So it's a very complicated process.
So you source it from yeast or from animal?
No, not from animal, from yeast. Yeah.
At the beginning, when you said about the sort of ancient kind of diets and how you think sort of activity lifestyles, how do you sort of know that? I mean, is that based on science? I mean, surely there's
No.
it was only so long ago before the actually
No, but you can,
expenditure.
when you look, for example, the indigenous people living, for example, in the rainforest of Brazil or in Borneo or something, you see how they live, and this is exactly the way they lived in the old time. They went for hunting. Let's say they go for apes. They hunt. They have really
Eat each other.
yeah. But they normally they went for collecting berries, because what else they had? They had no grains. They had no, let's say, vegetables they found in the forest or in the nature. They have not all the time. They have, let's say, fruits. So mostly they were depending on meat. What else cannot not imagine. And you see this when you look a little bit on these people, really Indians in the rainforest, they are still living like that.
Guys, Dr. Peter Koeppel.